was asked to give a presentation here, I was told this is a science talk, and I was like, well, I have to talk about Joe. And so I decided to talk about my science with Joe, which takes me from my first study to the last, which would be mere exposure and active avoidance. But I want to start, my story with Joe really starts with Mike Zaniga. I think Mike's not here. Oh, hey, Mike, how you doing? So Mike, as all of you probably know, is the godfather of cognitive neuroscience. And I came up in a time in graduate school when he was really working to sort of start this field, to get this field seeded with grant money and to get people cross-trained in cognition and neuroscience and computation. And Mike was instrumental in all of that. And I was thrilled when Mike approached me about doing a postdoc with him because he was the godfather of cognitive neuroscience. And I was studying at the time memory and amnesia as a graduate student. So my training was in cognition, but looking at patients with memory problems. But as I told Mike when he offered me this position, he was at Dartmouth at the time, and I'd spent my last year and a half of grad school in New York City, and I was in my 20s, and I was single. And I said to Mike, I love the job, I'd love to work with you, but I don't know if I can take Hanover, New Hampshire. And Mike said, well, try it for 6 months. If you hate it, you can leave. So I tried it for 6 months, I hated it, and I left. So that takes me to where Joe comes in. So that would be 1990 when I first contacted Joe about doing work with him. And I'd recently received my PhD, and I left a really good job, and I was unemployed. But I was fortunate in the sense that Mike was one of the few people that could really understand, just wanting to enjoy life. And he let me continue to do research, and we actually had some good publications together on memory and split brain patients. And he had enabled funding. He had convinced the James S. McDonald Foundation, along with George Miller, to fund postdoc grants that would allow people like me to cross-train in disciplines. So I was studying cognition and memory, but this would allow me to study, say, for instance, neuroscience. And given that I was unemployed, I had a lot of time to think. And I thought about studying memory, and I really did like studying memory, but I didn't like the way I was studying it, which was giving sort of neutral lists of words to people. It just didn't seem to me how memory worked in our lives. And so I decided at that point what I'd really like to do is study emotion and memory. And there just happened to be a great neuroscientist down the street, Joe LeDoux, who decided to take me on if I could get one of these grants. So I would go to Joe's lab, I'd learn about neuroscience, I'd learn about emotion, and I'd start studying emotion and memory. So that was the plan. I had to get the grant. So Joe and I came up with a grant. It had 2 points to it, 2 kind of main thrusts. So we thought, okay, what are the tasks on emotion and memory that we can do in animal models and in people? And one of them was mere exposure. So mere exposure means just the more you're around something, the more you like it. So Bob Zients had published in Humans, that essentially this effect existed. And unbeknownst to me, but Joe knew this, that effect had also been shown in rodents. So we thought, okay, one study we could do is do mere exposure, and we can do it in rats, and we can look at the effect of amygdala damage in rats. And whenever I figure out how to study the human amygdala, I can do it in people. And see if the amygdala plays a role in that sort of emotional learning paradigm. The second one, obviously, was fear conditioning, and I put threat there, but we called it fear conditioning then. And this was based on both work by Joe, showing, of course, the amygdala was critical in fear conditioning in rodents. And also, at that point, Arnie Ehrman was developing good paradigms that we can mimic in humans to measure, to do fear conditioning in human participants and measure skin conductance responses. So we could take Arnie's work and then look to see if we could see evidence for the amygdala playing a role in fear conditioning in humans. So I got the grant on the second try. Woohoo! And so it was 1991. I joined Joe's lab. It wasn't like Joe's lab today. Joe was still a relatively young scientist. I didn't get an office. I got a space at a counter and a drawer. But I took all the graduate neuroscience classes. That was my plan, was to really learn neuroscience. And I discovered that studying the function of the human amygdala is way more challenging than we anticipated. So fMRI was not a possibility at the time. For those of you who don't know, the first fMRI paper was published in 1991. There was one that year. Looking for patients with amygdala damage was difficult. I'd studied patients with hippocampal damage, amnesia, for a long period of time, but patients with amygdala damage who didn't also have severe memory impairments and hippocampal damage were very, very rare. And I would go try to talk to neurologists as a postdoc, and they didn't want to pay attention to me. So we start with the one rat study that we could do, which was the mere exposure study, and I apply for 3 faculty jobs, and I get 1 interview. And I should say, there was only one documented case at the time of a patient with bilateral amygdala damage, and it was this 1989 study from the Damasio group showing this Urbach Vita patient that showed, and the main finding was she showed normal skin conductance responses. And that was the only thing I could find at that point in looking at patients with amygdala damage. I know it sounds like it was a zillion years ago. It was 30 years ago. But we started with the fear conditioning study. So there was a study done, I think it was 1967, in rodents where they showed that they could show the mere exposure effect. And the way this study worked was they gave rodents they pre-exposed them for long periods of time to either Mozart or Schoenberg, another composer. So I'm just going to play for you briefly the kinds of things that they heard, these rodents. Let me see if I can figure out if I can, oopsie, I didn't want to get that back. Okay. This is Mozart. Should be familiar. Oopsie. Okay, how do I turn that off? Nope. I'm doing this wrong. This is my problem, I don't use PCs. Okay. All right, this is Schoenberg, in case you don't know him. I didn't know him. Now I really want to get that off. Okay. So what this study showed in 1967 was you could get mere exposure with rodents, but only for the rodents that heard Mozart. It didn't work for the rodents that heard Schoenberg. And so if you looked at the paper, right, it said, if you looked sort of down here, the Schoenberg, the behavior of the Schoenberg subjects posed a problem, and that, maybe it was because it was too complex, and that, that these behaviors require some level of stimulus complexity appropriate to the developmental and experiential level of the organism, et cetera, et cetera. So we thought, okay, well, so we can't use Schoenberg and Mozart, so we're just going to do simple tones. We're just going to play one tone for a period of time and another tone, see what happens. Now, Joe had a small lab at that point. He didn't have a lot of experiment rooms. No one wanted me playing tones in the lab all day long, and their rooms weren't available, so I had to do my experiment in the middle of the night. So I would come in every night. I would put rats in one of the rooms, play the tone, like one tone for 4 hours. I'd sleep on the floor. Then I'd play another tone for go downstairs, get more rats, bring them up, another tone for 4 hours, come back up, did this 3 or 4 weeks. And I discovered that, there's no mere exposure effect in rats, not surprisingly. I think we could have known that. And actually, I further discovered it's not so robust in people either. So, I learned a lot in the neuroscience graduate classes that year, and I got a job at Yale. My one interview, joined the faculty the next year. So I wasn't really in Joe's lab that long, and I didn't actually get much done with Joe. But fortunately, the James S. McDaniel Foundation agreed to let me use my funds that they had, leftover funds from my postdoc, at Yale to continue the projects with Joe. And Kevin Labar joined the project. He was a grad student at NYU, and he really kind of made this thing happen. So I get to Yale, and now all of a sudden neurologists will talk to me about collaborations. And they work with me to identify patients with amygdala damage who don't have problems with episodic memory. And these are patients with unilateral temporal lobectomy, a very common surgery for the control of epilepsy. And then Kevin also, worked very hard to develop fear conditioning paradigms in humans. We didn't have all this sort of push and play equipment. He had to do a lot to get that going. And we finally published our very first paper, which is 1995, 5 years after we started working on this. Essentially showing, taking patients with unilateral amygdala damage, presenting them a blue square, pairing it with a mild shock to the wrist, also having a control stimulus, a yellow square never paired with shock, and then measuring skin conductance. And here, what I've plotted is these first few trials are habituation trials. Here's acquisition, so you start to pair the blue square with shock. This is the differential skin conductance response, so the stimulus paired with shock versus the one never paired with shock. Control participants show, they show evidence of acquisition. The temporal lobectomy patients don't, and then we extinguished the response. So stop presenting shocks. So here, we also did this. We also were able to identify one rare bilateral amygdala patient. There are not a lot of them out there, but the neurologist at Yale had a woman who had bilateral amygdala damage that they wanted me to get to know, because they wrote me saying, what does the amygdala do? And I'm like, I don't really know. But can we study her? And they said yes. So here's our sort of a cartoon version of our paradigm. Here's a cartoon version of the data for the amygdala patient. So this would be a healthy control participant. Here's the shock. Here's the skin conductance response. Here's the blue square that predicts a shock. And what you can see is they see this anticipatory response, not surprisingly. Here's a patient with amygdala damage. They show this normal unconditioned response. They show skin conductance response to the shock, but not to the blue square that predicts a shock. Here's a woman with bilateral amygdala damage. That's Kevin LeBar in the background. And she was shown her real version of her data with fear conditioning, we asked her what she thought. So I'm going to play that for you because it leads to a point I want to make, if I can get to work.
I haven't noticed that there's anything odd about my reactions to normal lifestyle, life threats. My fear element seems to be, to me, in my head, normal. I've never noticed anything odd.
And how do you feel seeing these results? How does that make you?
I'm dead. I think that it's probably absolutely correct. I know I don't sweat. I knew that there was an anticipation that the blue square was going to, at some particular point in time, bring on one of the volt shocks. But even though I knew that, and I knew that from the very beginning, except for the very first one where I was surprised, I really don't think that I mean, I knew it was going to happen, but that was my response. I knew it was going to happen. I accepted that it was going to happen. So I learned from the very beginning, it's going to happen. Blue shock happened. I turned out to be right. It happened.
So, as she somewhat dramatically demonstrates, in spite of the fact that she fails to show me this physiological evidence of threat conditioning, she has perfectly good knowledge of what happened. And when you talk to these patients, they didn't seem odd in any ways. It doesn't seem, we know that they didn't show fear conditioning or threat conditioning, although we called it fear conditioning then. But at the same time, they seem to have relatively normal lives. So we actually did diary studies with this particular woman and just sort of one-shot studies with unilateral patients and showed that they showed the normal range of subjective experience of fear and anxiety. And I bring that up just because this is a point that's come up many times, but this was even around the year 2000, we were able to show that there's this dissociation between the defensive responses and the subjective feelings of emotion in these patients. So now I'm just going to start to go through like all the other studies we did. So I'm sorry if I bore some of you, but I just really needed to talk about what I did with Joe. So we had a little bit more success in 1998. We had our second paper. So fMRI became a reliable tool to study the human brain, and Yale, fortunately, was a leader in this technology. So fairly early on, I was able to work with a great physicist who taught me about functional imaging. He made me do two other experiments I didn't want to do before he let me do the one I want to do, which was this one. But nevertheless, we got it done, and we were able to do the first functional imaging study showing activation of the amygdala to a stimulus that predicts a shock relative to one that does not. But if you know anything about imaging, you know our analysis was quite different than the way you do it today by looking at this. And then, in 1999, I was at Yale, so I had to leave because they didn't tenure people back then. I leave Yale and I joined the faculty of NYU. And there I oversee the design and the construction of the first brain imaging facility, and I get my first postdoc. And then Joe and I got to continue our collaboration much closer than we were previously. So when I got to NYU, most of the work we've done since I've been there, and I'm going to say to now, has been not so much looking at kind of the acquisition of threat reactions, but really what it is we do to control them, particularly when they're maladaptive, when we want to extinguish them, when the stimulus no longer predicts the shock. And the very first study we did, I think Lorenzo gave you a nice background. Joe was one of the first to show a role for the prefrontal cortex, and particularly the infralimbic cortex, in extinction learning. And so the very first study we did, which is our most highly cited research publication to date, was to look to see if we see evidence for the ventral medial prefrontal cortex involvement in extinction learning in humans. So I've put here because I want to make this point, I've put here the skin conductance data. The dark line here is the stimulus that's paired with shock. The light line is the stimulus never paired with shock. This is during acquisition, so we see a difference. We did a session of extinction immediately after acquisition, so we just stopped presenting the shock with the conditioned stimulus. And then here we saw a lot of spontaneous recovery, because I'm going to come back to spontaneous recovery and then further extinction. So, perhaps not surprisingly, we found what we would expect from the animal work of Joe, and also at that point Greg Quirk as well, with Joe and both independently from Joe, that we see sort of decrease in amygdala bold signals consistent with inhibition of the amygdala during extinction learning relative to acquisition, which is here in red. We see this region of the ventral medial prefrontal cortex that we think is analogous to the infralimbic cortex. This is characterized by a decrease in bold signal that diminishes with successful extinction learning, and the sort of change in activation of this region predicted the sort of retention of extinction the second day. So soon after that, around that same time, Joe and I started to work on a review article. We were near each other, and this was sort of our one global review article. It is my most highly cited paper, and it is also our most highly cited paper. It is not Joe's most highly cited paper. And it was a fairly comprehensive review. I don't know if that would even be possible to do today, Joe, given all the things we'd want to talk about. But it's a paper I'm very proud of. So then we then moved on to, so the rest of the studies that we tried to do, and sort of from now till everything I'm going to talk about till the very end, has tried to say, okay, we know we can extinguish responses, but how is it we can have more persistent reductions of threat reactions? So there was a finding out by Michael Davis, who suggested that if you did extinction right after you learned something, right after acquisition, then that extinction is very permanent, so you don't get things like spontaneous recovery or reinstatement. Now, we had already had some data we'd published previously in another study that suggested that wasn't the case, but Daniela came and Chris came, and they got together and they're like, we're just really going to make this point really strong so that we can tell Mike he's wrong. They didn't do it because of Mike, but So Daniela reinvigorated a study that I'd done previously with Kevin Labar, where we did contextual conditioning, and VR wasn't really as easy to use back then, so we actually took rooms and made them as different as possible and wheeled our subjects back and forth between them. And what we knew from other work is that reinstatement is contextually mediated. If you get a reinstatement cue in a different context than the one with extinction, you're more likely to see a bigger return of the threat response, and that's exactly what Daniela found. So here's acquisition. The white bars are the same context. The black bars are different context. Here's acquisition. Here's extinction. Here's the last trial of extinction. And here's the first re-extinction trial, either in a different context where we have a lot of reinstatement, or in the same context where we have less. So again, even though it was immediate extinction, we saw very good recovery. She found the same thing for just spontaneous recovery, another paradigm. And here's one of the few studies where we actually published the human and rat work in the same paper. So this was a study by Chris Kane, looking at rodents, essentially doing the exact same thing, immediate extinction, or also looking at delayed extinction, and no difference in the post-extinction test, either for reinstatement or for spontaneous recovery. Another question that we'd often had is, what is the relationship between kind of the things that humans do to control our threat reactions and what you can do in other animals? So, humans, we can use our thoughts to control our emotional reactions. We can regulate our emotional responses. Other animals, we can think about extinction, where you present things and you eventually learn the stimulus is now safe. And in our work on threat acquisition, we were able to show that, even though we have this phylogenetically older system for threat learning, for learning the association between a CS and a US, if I do some type of social intervention, like just tell you now the threat predicts a shock, I will also engage the amygdala. So in other words, this phylogenetically older system was co-opted by human knowledge, in this case linguistically conveyed threats, to express the threat reaction. And so we asked the question, what's the relationship between the circuitry of extinction and the circuitry of emotion regulation? Could it be that using our thoughts, using our cognitions to change our emotional reactions actually co-ops the mechanisms of extinction to reduce emotional responses? And so we did this using, again, another threat conditioning paradigm, but this time participants, prior to seeing either the stimulus paired with shock or the one not paired with shock, were either told to attend to their natural feelings when seeing these stimuli, or to regulate their emotional response by imagining a scene from nature that was soothing to them. And using this sort of cognitive strategy, here, participants were able to reduce their skin conductance response to the stimulus that predicted shock. What we found in the amygdala was when people were using this strategy, they were also able to reduce their amygdala response. And in the prefrontal cortex, we saw two regions that showed greater activity when you're regulating versus attending. One is the lateral prefrontal cortex, and this is a region that has been implicated in emotion regulation to be involved in the executive control needed to think about the stimulus differently. And the other was this ventral medial prefrontal cortex region that was overlapping with the regions we saw engaged in extinction learning. And if we looked at this region, here I've plotted the data from our extinction study and our emotion regulation study. Here's this decrease in bold signal we see in early extinction that goes away that increases as you extinguish your conditioned response. Here's when you're attending to the emotional significance of the event or regulating. And if we took sort of the region of interest from our extinction study, this ventral medial prefrontal cortex region, and said, show me regions that are correlated with responding when you're now using your thoughts to regulate your emotional response, we find the amygdala and the dorsolateral prefrontal cortex. So just to wrap up those, just to sort of give you the take home from that particular study, what this suggested to us is that, we know the amygdala from the work of Joe and many other people, but mostly Joe, was important for the acquisition, storage, and expression of this simple associative threat memory. When you now extinguish that threat memory, the ventral medial prefrontal cortex inhibits the amygdala to allow the expression of the extinction memory. And when you use your thoughts to regulate your emotional response, the dorsolateral prefrontal cortex now co-opts that circuitry of extinction so that your thoughts can now also diminish your defensive responses. All right, and that was work by Mauricio when he was in the lab. So I'm going to go through a couple of these quickly, just because I don't want to give you all the scientific details in the world, but this was a study that Daniela did, again, trying to say, how can we flexibly move around threat responding? And so what she did, instead of doing acquisition and then extinction, she then said, let's make it even harder. Let's now have you acquire one stimulus predicts a shock, the other predicts safety. Now we're going to switch it. So now the stimulus that predicts safety predicts the shock, and the stimulus that predicts the shock is going to predict safety. And what she did when she did this was she found, so here is the behavioral result. So plotting the stimulus that predicted shock first from the one that predicted shock second. Here is here's before the reversal, here's after the reversal. So people acquire their conditioned response, and they switch their conditioned response. Here's the ventral medial prefrontal cortex, the same region we see in extinction. We see this decrease in that region that we see during acquisition. It starts to go up, but unlike in extinction where it sort of just goes to baseline, it actually goes above baseline. So in other words, it's working harder a little bit when you have to now not only inhibit that one learned emotional response, but also learn a new one at the same time. With the amygdala and the striatum, simply track the skin conductance response. And another technique that Joe was starting to look at more at that time was avoidance. So Danielle, I think, showed you this slide earlier, suggesting that when there's a separate pathway for when you learn to avoid a conditioned response that goes from the lateral nucleus to the basal nucleus to the striatum, as opposed to when you passively respond, which goes from the lateral nucleus to the central nucleus to the brainstem. So we tried to do an avoidance study. I have to say this was our first stab at this. I'm going to finish with avoidance as well, where we had 2 stimuli, one of which was paired with shock relative to safety stimulus, another one which was paired with shock, but then people had the opportunity to avoid the shock. And what we found was that the skin conductance and the amygdala response tracked whether or not it was paired with shock or you could avoid. And we found slightly greater striatal responses when you could avoid the shock versus when you could not. So then this takes us to what Danielle, I think, has already talked about some, which is this sort of this research on reconsolidation. So Joe in 2000, has been talked about already, with Kareem Nader, showed that we could persistently inhibit threat responses or consistently change threat responses by targeting reconsolidation. And of course, we couldn't do the kind of invasive techniques that Kareem did in humans, although we talked about what we could do. We tried different things, it didn't work. And so Marie Monfils and Daniela Schiller discovered this behavioral intervention. So essentially, if you can essentially reactivate the memory prior to extinction learning, essentially start the reconsolidation process prior to extinction learning, you show evidence that you don't show the recovery of the threat responses at a later time. So I don't want to walk through this in too much detail, except to say, this was one of the few papers where we actually tried to submit papers back to back from Joe's lab and my lab. Marie Monfils' papers in rodents, our papers in humans. We submitted back to back to Science. Marie's got into Science, ours got rejected for the primary reason that fear learning, I guess, in humans is not a good model of human fear learning. The idea was, we can't really do very intense manipulations in humans, so therefore, our results aren't as compelling as the rodent work. But there was an upside to this. We then went to Nature. It took a year to get Daniela's paper into Nature. And in that year, she was able to bring some participants back and show that this effect lasted a year later. So in other words, she could reinstate the threat memory in participants who underwent standard extinction, but no evidence in participants who underwent safety training during reconsolidation. And this was followed up with an imaging study. And in this imaging study, I'm just going to give you the take-home message from this. Daniela showed that when you have one stimulus that is undergoing standard extinction training, you get what you expect in the ventral medial prefrontal cortex, which is this decrease in bold signal that diminishes with extinction training. But if another stimulus at the same time, simultaneously, is undergoing safety training during the reconsolidation window, you get essentially no change in the ventral medial prefrontal cortex. And you get much stronger connectivity between the amygdala for a stimulus undergoing standard extinction training here in black than one that's undergoing safety training during reconsolidation here in orange. And finally, Annemieke Perjes-Schoute also followed up on a study from Joe. So Joe, we've done these studies showing that we can diminish the amygdala response with extinction. We can diminish the ventral medial prefrontal cortex response with extinction. But Greg Quirk a while ago in Joe's lab showed that there's also changes in the auditory cortex when a tone gets paired with a shock. And those tone-specific changes persist even when the conditioned response no longer persists. And so Annemieke was interested in seeing if she could find evidence in humans that the auditory cortex response persisted even when the conditioned response no longer persisted. And she did. So at the end of extinction, when we're no longer seeing conditioned responses, there's no response in the amygdala, no response in the auditory thalamus, no response in the primary auditory cortex, but a big response to the CS plus relative to the CS minus, the stimulus paired with shock versus the one not, in the auditory association cortex, and no response in the medial prefrontal cortex. 2 more studies. I have 5 minutes, you said? No? Okay. So this is one of my favorite studies only because it was one of the few studies where we did it in humans and then we went back and did it in rodents. So this is a study where during extinction learning, we try to present a novel stimulus to reduce uncertainty. And what we find in this case is little evidence of threat recovery, and we find the same thing in rodents. And it's one of the few times I've been able to say to Joe, hey, we got this cool study in humans, can you try it in rats? And we can find the same thing. So now this gets me to active avoidance. This is the final study I'll talk about. It's our most recent publication. So, one of the things that Joe showed with Chris Kane and later with Jessica Mascarello, that if you give a rodent an opportunity to avoid a shock or avoid a CS, what you find is not only, that you can, the rat can learn this, but also that you find less evidence of spontaneous recovery or reinstatement, or less evidence that the threat response comes back. So in other words, it's almost like building a resilience. You give an animal control over a shock for a period of time. Even when there's no longer control, you show less evidence that the threat response returns. So we were interested in finding, can we find evidence that, allowing active avoidance attenuates Pavlovian condition responding in the future. And so we used a paradigm similar to what Daniela talked about earlier, where we had sort of this shuttle box where people could learn to turn off the shocks. We had yoked participants who were matched to individuals who could turn off the shock, but they never could. And then we looked to see whether the threat response returned the second day, and they also now learned a new threat conditioning association. And I'll just go here. So what we found was we reduced spontaneous recovery with this avoidance learning, and we also reduced new threat learning with this avoidance learning, consistent with this persistent control. And Joe would show that the ventral medial prefrontal cortex, or Justin Mascarello and Joe's group, was important in toggling between this pathway that does a passive response versus this active response in the striatum. And so we also saw greater prefrontal cortex activity in our individuals who underwent avoidance versus extinction, and also greater striatal response. So that takes me to today. So Joe, I was looking, I thought we had more publications, but we only have 18. It's not, So Joe and I have had 18 publications to date. We have 9,179 citations, and we have an H-index of 18. Our papers are highly cited when we do them together, so maybe we should do more. But I just want to say that's sort of and counting. I'm now just accepted a job at Harvard University, so I'll be leaving NYU. I won't be around Joe, but I'm still planning on working with Joe and collaborating with him. And I just want to say, Joe's been my best collaborator, my biggest support, and the most influential scientist in my career. I am forever grateful, and thank you, Joe.